Hello everyone. Today I will present my work on MRI guided hierarchical sectioning and stitching of the brain tissue blocks for precise alignment of histology images to corresponding MRI volume. So as part of the study, first I'll present about improved ex vivo imaging using brain and container specific 3D printed holders. So we know ex vivo imaging allows to increase resolution and SNR due to the reduced time constraint, and it's used in allows validation of in vivo imaging modalities and also in registration of histology to in vivo MRI. Two main factors that have direct impact on the quality are an RF coil that provide uniform sensitivity and high SNR, and systematic placement and stability of brain inside the container during imaging. To obtain high quality ex vivo image, we propose using container specific and brain specific 3D printed holders designed using preliminary data. The preliminary scans of the container are used for extraction of the container surface and identifying optimal positioning within the container. We propose a design that enable imaging several brains in separate sessions while positioning them consistently in the same position relative to the RF coil. And for swift model creation, we have developed a semi-automated software pipeline for creating a 3D printable model. So to do this, we acquire MRI of the container filled with salted water, and we created the inner surface of the container and identified optimal position of the brain to be in the container to be placed during the imaging. We also acquired a low resolution MRI of the brain in fluorinator. Using the images acquired, we create the surfaces of the brain and the container, which will be used for the model creation. Using the two surfaces, we create 3D printed holders with profiles that fit the surface of the brain on one side and the inner surface of the container on the other. In optimal position, the specimen is in a region of uniform sensitivity and high SNR. We identify the optimal position using in the container using SNR and estimated bias field map acquired in the preliminary scans. Here's a setup with fluorinator as immersion medium. The white structures here inside the container is the 3D printed holder, and it holds the brain like this. It supports the brain during imaging. So the outline of the pipeline itself, for creating the holder, the user can provide MRI of the same brain to be imaged at a higher resolution, or a template which is scaled to match the dimensions of the image to be imaged, brain to be imaged, sorry. In general, the model creation involves following steps: a reorientation or rotation, brain extraction or morphological operations, and volume extrusion in specific direction, and surface generation, and also model creation while incorporating the information from the container image and also the surface and which will be 3D printed. So pipeline provides multiple design options other than what I've shown here. User can choose what suits their needs. So here I show image acquired without the brain specific holder, and an image acquired with the container and brain specific holder. The image obtained without the brain specific holder and with no consideration to optimal positioning shows the bias field, as can be seen here in the when comparing the dorsal and ventral part of the brain. Whereas the image acquired with optimal placement using the container and brain specific holder show uniform signal throughout. So now I will talk about my project on MRI guided hierarchical sectioning and stitching of tissue blocks for precise alignment of histology images to the MRI volume. Doing so, I will highlight how I have used ex vivo imaging. So the motivation one for this work is the spatial resolution of the ex vivo MRI. High resolution ex vivo MRI is beneficial for registration of histology. And then acquiring high resolution MRI in the range of 100 microns for human and macaque whole brain is difficult using a large bore scanners with gradient strength of 80 millitesla to 80 millitesla per meter. The preclinical scanners have, whereas the preclinical scanners have powerful gradients around 300 to 400 millitesla per meter, but small bores that cannot accommodate large brains. Motivation 2 is the advantages of small slices. What do I mean by that? Histology of large brains, such as human and macaque brains, is challenging due to tears and distortions, as you can be seen here in the example image that I've shown here, and requirement of specialized instruments, as have been used in previous works, as shown here. So the solution is to section the brain into blocks of manageable dimensions, and high-resolution imaging of the section tissue blocks can be performed using ultra-high field small bore MRI systems, and stitching the blocks to form high-resolution image of the large brain. Here is the flow chart of the hierarchical sectioning and stitching procedure that we have developed. There's a lot to unpack here, but the dark arrows represent the chronological order in which the processes are performed. So we have, and the red arrows and the green arrows show the information flow between the stages. For example, the information from the cutting locations are used in the stitching stages. So let's go in the order. So in the imaging stage 1, that's where we acquire the ex-vivo MRI of the whole brain, and which will be used for in the sectioning stage 2 to basically to plan the cut locations based on the instrumentation requirements. In the later stage, we image the section blocks at a high resolution using the small bore scanner. And to provide an example, this can be sectioned further, as we have done in the posterior section here, and imaged further again. And these blocks are then processed for histology. And for the reconstruction process, you could basically, we are stitching the images acquired in the stage here, and subsequently for the larger blocks, and then they are registered back to the in vivo MRI. So we pre-plan the cuts according to our instrumentation requirements. In this case, we plan the cuts based on the cassettes used for histology processing and paraffin embedding for microtomy, while also considering the dimensions of the RF coil and the container to be used in imaging. Initial ex vivo MRI is shown in the panel A, and panel B shows the planned cassette locations for this study. So the first row shows the planning of the cut locations for the sectioning stage 1. The red arrows highlight the planned cut locations, and using a 3D printed sectioning model specific to the hemisphere, we cut the hemisphere into 4 blocks as shown here in panel B. And panel C shows high resolution images acquired using the preclinical scanner. The blocks are imaged separately, but shown together for simplicity here in this image. Similarly, we could repeat the procedure for the posterior block. Just as an example here, we have sectioned the posterior block into 3 slabs, and the 3D printed model, the planned cut locations are shown in the panel B. And with the 3D printed model, the design of the 3D printed holder model is shown here in the panel C, and the sectioning itself shown here in the panel D. And the final high resolution ex vivo, the image acquired using the small bore scanner, is shown in panel E. The slabs are imaged separately, but shown together here for simplicity. So from the imaged high resolution slabs, surface features and extracted features from the volume, high resolution MRI volume, can be used for the registration. Here I'm showing the surface, segmentation, and normal vectors, which will be used to drive the registration to stitch the adjacent blocks. Just to explain the methodology here, the stitching methodology here, this outline shows how the linearly registered slabs are stitched together using a surface registration method. So as mentioned, the surface registration uses features shown in the previous slide, and the stitched surface, the final stitched surface, is shown here. The deformation from the surface is then applied to the volume, and the final merged volume is shown here. In the end, there's another final nonlinear registration after stitching all the slabs from this stage. it is nonlinearly registered to the posterior block, which is used for planning the cut locations. So here are the results from the stitching stage. So the panel A shows the high resolution ex vivo this MRI of the slabs, and panel B is the posterior block used for planning the cut location creation of the 3D printed model, which will be used as a reference for the registration itself. And step one is the linear registration, results from the linear registration. Here you could see the overlap between slabs and small gaps in between the slabs. In panel D, it's the result from the surface registration. You can see that there are small inaccuracies, but the gaps and the overlaps are pretty much gone. And then the panel E shows the final nonlinear registration, which pretty much aligns the slabs, the stitched slabs to the posterior block, which is used as the reference. The same approach can be adapted for the larger block, is adapted for the larger blocks as shown here. And by concatenating the transformations and displacement fields from all this steps in a stage, and also from all the stages, you could basically transform the slabs from its high resolution space to the original whole brain ex vivo space as shown here. And panel D shows the slabs put together along with the blocks. So in the same way, the chain of transformation and deformation can be applied to the reconstructed histology volume after registration to the corresponding slab. I'm showing a representative registration of histology to the corresponding MRI slice. So the histology space is on the top and the MRI space is on the bottom row. Here I'm showing a Nissl stained histology slice and manually delineated gray matter white matter boundary, and the low pass filtered grayscale image, which is used for registering to the MRI shown here. So panel E shows the histology registered to the MRI along with its edges, and panel F shows the MRI with the edges from the histology overlaid on it. So we present a semi-automated pipeline for MRI-guided hierarchical sectioning and stitching of fixed tissue blocks for improved alignment of processed tissue image to the corresponding MRI images. So the methodology is, oh, sorry, I switched orders. So to conclude, the improved ex vivo imaging using the brain and container-specific 3D printed holder, it's a methodology for obtaining high quality ex vivo image. And the method is applicable for human brains as well as the brains of animal models. In terms of the MRI-guided hierarchical sectioning and stitching of the brain tissue blocks, our method allows alignment and stitching of high-resolution MRI of brain tissue block for reconstructing an MRI volume of the full brain at high resolution, and precise alignment of images of artifact-free processed tissue to corresponding high-resolution MRI volume of a full brain. So I would like to thank my supervisor and some people from our lab and at the MNI who have contributed to this project. Thank you very much, and I'll take questions. Thank you.